Hello everyone, my name is Shujat Hussain and today we will talk about the advanced doping. So in this presentation we will see what is doping, what is technology doping, and what is gene therapy, what is gene doping, and the potential genes that can be used as doping and why it's controversial. So let's start. In 2011, an anonymous survey asked more than 1,200 athletes whether they use performance-enhancing drugs. The results kept quiet for the 6 years, and the results were so much shocking. Like the 44% admitted to doping, and the rest, the 56% didn't admit it to doping. So, but on the other hand, in 2017, the when WADA shared the samples of tested positive for the doping were only 1 to 2%. The tests were positive. So, what is doping? Doping is the use of prohibited medication, drugs, or treatment by athletes with intention of improving athletic performance is known as the doping, like anabolic drugs. Anabolic steroids, the drugs helps build muscles, tissue, and increase the body mass. The stimulants, some substances like cocaine and Ritalin and amphetamines and caffeine, nicotine, used by athletes to reduce fatigue and increase their alertness because it helps to boost their CNS or the central nervous system. The hormones, the hormones are responsible of every bodily function, including muscle development and muscular growth. The- As some athletes use hormones to increase muscular mass, but as the muscular mass increasing, it's having some side effects, which having some negative effects. The oxygen transport, or it's also known as the blood doping, and the oxygen transport means the increasing the oxygen uh, carriers like the red blood cells, increasing the red blood cells in the human body. So it is the main source of oxygen transportation to till the cells, so it increases the uh, endurance as the oxygenation process increased. So some athletes do blood doping, or some like they extract their blood 2 3 months before the event, uh, during the event or the before the event, it's very short time they inject the blood into the soil, they increase the blood volume. And some athletes use the erythropoietin, the EPU, so it also increase the RBC, the red blood cells, so the oxygen carriers are increased, so it helps in the aerobic process and increase the endurance of the athletes. So what is technology doping? Improving the athlete's performance by using the substances is the doping, and improving the equipment's performance using the hidden technology of the devices to increase the performance of athletes is known as the technology doping. For example, the LZR racer swimsuit, vaporfly shoes, ionized shirt, mechanical doping. So these are the LZR racer swimsuit was used in 2008 Beijing Olympic Games, and that suit worn by the athletes, and the athletes are that year in 2008 about 23 world records were broken in out of 25 world records, and all the athletes who broke the world records were wearing the LZR racer swimsuit. So the Vaporflies. After that, the FINA has banned the use of full body LZR racer swimsuit. And the Vaporfly shoes is also made by the Nike in 2007, which also helps to increasing or the helping in increasing the speed during the running, and the ionized shirts and the mechanical doping. The mechanical doping is used by the cyclists in 2010 in Tour de France. A cyclist used a very small nano battery and having the very small motor, which is giving him the very little support to a very little assistance or the help in increasing the speed of the cycle. So this is the examples of technology doping. What is gene therapy? So gene therapy refers to the replacement of faulty genes or the addition of new genes to cure diseases or improve the ability to fight against the diseases. The first clinically tested in 1990 for the treatment of diseases. Gene therapy was used there. In 1999, Jesse Gelsinger first died in clinical trials suffering from the inflammatory reactions. In gene therapy, there are two different methods used in gene therapy. The first is the ex vivo, and the second is the in vivo method. The ex vivo gene therapy method is the process of genetically altering a person's outside of the body and then transplanting them back into the human body. So the ex vivo method uh, and after transplanting or planting the advanced or the uh, uh, required functionality or the functioned gene transplanted into the human body, so body will work according to the newly gene instructions. The in vivo method, the in vivo gene therapy refers to the direct administration, known as the systematic administration, or locally to a specific organ of the interest, like to the brain tissues, and to the muscles, to the eyes, to the testicles, to the kidneys, directly injection to the human body organs or the systems. What is gene doping? And the non-therapeutic use of cells, genes, genetic elements, or gene expression modulation capable of improving athletic performance is known as the gene doping. In 2003, WADA proactively added gene doping to the list of banned doping practices. The Office of Drug Control Policy Reauthorization Act of 2006 permanently includes gene doping in the list of prohibited substances among any list adopted by the US Anti-Doping Agency. The potential genes that can be used in doping: the EPO erythropoietin, highly used in gene doping. Target tissues of the system is the blood system. Physiologically, it increases the number of red blood cells and increase the oxygenation. As the red blood cells increase, it increase the oxygen carriers, the oxygen supply, sufficient oxygen supply, and it increase the endurance. So the risk associated with that is as the RBC increased in the bloodstream, so blood viscosity is increased. So difficulty in the laminar blood flow through the vessels. As the laminar blood flow become the turbulent flow, so it will damage to the blood vessels. IGF-1, IGH, the insulin-like growth factor 1, and or the immunoglobulin have it. The target tissue of the system is the endocrine and the muscular system. Physiologically, is the excessive growth of bones and the tissue mass, as the muscular hypertrophy and the hyperplasia, with the stimulation of glycogenolysis and increase the release of glucose from the liver, and increase the lipolysis, the breakdown of the lipids, and decrease the lipogenesis, and it increase the protein synthesis. Ultimately, the goal is the increasing the endurance, and it increases the muscular mass and the muscular strength. The risk associated with that, the it 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 it, abnormal the vision, headache, nausea, vomiting, and the athletes will feel pain in their joints, in their muscles, and the overgrowth of the cartilage in nerves and jaws, the insulin resistance, and the diabetes. HIF-1, the hypoxia-inducible factor 1. The target tissues are the target system is the blood system and the immune system. Physiologically, it works like the erythropoietin, like increase the RBC and increase the muscular strength and endurance. And the health related risks are the it increase the blood viscosity and increase the hypertension in the athletes. ACTN2 and ACTN3 as actinin alpha 2 and actinin alpha 3. The target tissues of the system are the muscular system. Physiologically, it increases the muscular mass, the muscular strength. The risk associated with that, currently no data found on negative effects of the gene doping using ACTN2 and ACTN3. Why is controversial? There are some objections against the use of the gene doping and gene editing or modification in the field of sports. One concern is that the use of this technology could result in unexpected consequences. The the new class of enhanced humans that would result from the germline enhancement would when interact with the unenhanced humans in the sports, thereby the creating a potential disadvantage for the it's a potential disadvantage for the unenhanced athletes, and this may cause the state of inequality between the athletes. So it is the next big threat to the sporting world, but currently no data or the uh, uh, no one has been detected as used or used for the gene doping, because the problem is that that this is still undetectable, but in future there will be time when it will be detectable and the athletes have the same penalties like having the same penalties like the other athletes who do doping. So the potential use of the gene doping is critically dependent on the increasing understanding of the genetic modifiers of exercise physiology and medical development of the gene therapy system. While while there is no evidence to date that athletes have practiced gene doping, so the threat of gene doping has led to many laboratories working to develop systems to identify and to detect any possible gene doping in the field of sports. Thank you very much.